Welcome to Bio 1104. In this lecture we're going to be continuing on with nutrition, growth and control of microbes, and hopefully when you do your practical sessions as well, this will come into a little bit more focus for you. Now we're going to be looking at the control of microbes, but the question is, why would we need to? Okay, hopefully by now you've recognized the population of microbes that are found in and on us and in our environment. Okay, so typically we need to control those particular microbes that can be pathogenic, and we might also want to control particular microbes, say in a food industry, that might be spoilage organisms. So we need to control them, and we can't just blanket kill all of them because, well, we're not going to be able to for a start, but you might be killing or retarding the growth of some microorganisms that you need. So, you know, what would life be without cheese or yogurt? So we need to think about the mechanisms of control. So how can we target particular types of microorganisms? And those control methods, how do they actually work? So we need to be aware of that. And so we're going to be looking at some of the methods of control, so physical, chemical, and those actions, the mechanisms. And also some of the limitations, because you have to be aware of, if something doesn't work, what could have gone wrong. So you're starting that critical evaluation, critical reasoning. So I want you to be able to talk about the rationale for controlling microorganisms in the environment. Okay, it might be on a human, it might be in a human, it might be in a wound, for example, or it might be in an environment, say in hospital. You want pathogenic microorganisms controlled in a hospital environment, don't you? Because obviously not many people who are very well go to hospital. So that means that their immune system is compromised in some way, so you don't want to be overloading it with possible pathogenic microorganisms. So we're going to look at some of the some terms: sterilisation, disinfection, antisepsis, pasteurisation, sanitation. Do you know what they actually mean? Sometimes we use them interchangeably, but there are distinct differences between them. And as I said, some of the mechanisms for controlling microbes. So if you think about food production, and that's probably close to everyone's heart. Yes? Okay. So if you have a particular food product being produced, you have to have a particular type of organism, a particular species to help with that manufacture, that production. Okay? Now, what if you had another type of microorganism in there as well that you haven't controlled for? That might cause food spoilage. So instead of being one flavour, it's quite a different flavour. Typically not so nice. It might be with juice. You might have contamination by yeast. And so have you ever seen in the cold section of the supermarket some fruit juices that have kind of swelled? Yeah? That's other bacteria growing in there producing gas, and that's why you get that swelling. If you've left your milk a little bit too long, you get separation. Yes? So that's overgrowth of certain types of bacteria. So some of these things, some bacteria are very useful, but some can be problematic. So food, everyone has probably seen that. But think about in a hospital, maybe a wound. If that wound is contaminated or infected by something, you want to be able to control that, because if it's infected, your body's going to be trying to get rid of the infection and not trying to heal. Okay, and there's a lot of other things that would be happening at the same time. But you certainly don't want those opportunistic pathogens. So that's what we can use. What we can use to limit those contaminating organisms are chemical means and sometimes even physical means as well. And another type of chemical means is antibiotics. So you've probably all had a course of antibiotics in your life, at least one. So how do antibiotics actually work? If you have a sore throat, a virus, if you go to the GP, do you get antibiotics for a virus? No, because antibiotics only work on certain bacteria. Okay, so it needs to be specific for the type of bacteria or microbe. So some definitions. Sterilization is a process of destroying all microorganisms. Okay, so you get rid of them all. And that can be through steam, through heat, sorry. Bless you. So heat, and that can be steam with pressure, such as an autoclave, where you have to, you get steam at 121 degrees Celsius with pressure, and that for a certain amount of time, and then that will give you sterilization. And we'll talk about some of the checks on that as well. Gas can also be used, certain types of gas can be used for sterilization, such as ethylene oxide. Now, it's great. You have used the Petri dishes in prac. They're those plasticware. Obviously, you're not going to be putting them through a heat system, but those Petri dishes are sterilized by ethylene oxide, typically. Now, terrific, they don't distort the plastic or anything like that, but ethylene oxide is really toxic. So you have to spend the thing to be sterilized has to remain in that environment for probably 8 to 10 hours, and then you have to get rid of the gas somehow. If it goes onto your skin, it's really corrosive. Okay, so you need to be very careful when you're using that sort of thing. Why do you use sterilization in the food industry? Obviously, if there's a foodstuff that's in a can, you want to sterilize that. You want to destroy any endospores, because if you think about the environment in a can, it's anaerobic. So if there are any endospores in there, that the heat treatment during the process might be enough to kill off all the vegetative cells or the living cells, but the endospores might survive. And then if that hasn't been sterilized, you get to, maybe it goes to a certain temperature, and that might be enough to germinate the endospores. And they produce toxins and gas, etc., etc. So that's when you can get spoilage of cans as well. And Clostridium botulinum endospores are notorious for surviving some sorts of heat treatment in cans. Disinfection, it is a process of killing or inhibiting microbial growth on a surface, a non-living surface. That's disinfection. But the opposite of that is, or the corollary of that is antisepsis. So that is kill or inhibiting microorganisms on a living surface. So disinfection for non-living surfaces, antisepsis for living tissue. Okay. Now, just a little bit of a caveat to that. If those surfaces have a build-up of organic material, choose your own type of organic material on that, that disinfectant won't get to the surface covered by the organic material. So to be effective, it needs to be a clean surface. Yep. Pasteurization, back to milk. This is where, actually, and other foodstuffs, you get brief exposure of a high temperature for a particular time. So it could be 63 degrees Celsius for 30 minutes or 72 degrees for 15 seconds. Why would you use different times? Well, that flash heating might be enough to decrease the population of microorganisms, but not enough to denature the foodstuff. Okay, so it's always a balancing act between decreasing the population and changing the consistency or the organoleptic properties of that particular food. So you have to be aware that this is not killing the microorganisms, all of the microorganisms. It's actually just reducing the load. So if you've got something that you're going to pasteurize that has got a high microbial load, you're going to only decrease it a certain amount, a certain log number. Okay? Sanitization is what you do to, say, things like food utensils, to decrease the microbial counts. Hopefully the right type of microbial counts are going to be decreased, or the right kind of microbes, and on utensils. So you want to kill off hopefully the ones that might cause food poisoning or adverse effects. Few more. A bactericide is anything that kills a bacteria. So if you have a suffix which is CIDE, so a side, that means that it kills. But a bacteriostat, or something that is bacteriostatic, will only inhibit the microbial growth. Okay, so you just have to be aware of those changes. Now, I've just thrown in sepsis as well, because if you don't decrease the population of bacteria and you get an infection, then that infection can go into the blood and you get sepsis. Now, sepsis is the presence of bacteria or their toxins in the blood and the effect on the body, okay? Because your body's going to try and fight that particular infection. So, if we look at the effectiveness of antimicrobials, so they're not all the same. So, if we look up at the top, these are the most resistant to antimicrobials, and these down the bottom, these are the least resistant. Okay, so prions, everyone's heard of prions? Yes, they are the hardest to get rid of. Okay, then that's followed by endospores. They are very difficult to get rid of. And as it goes right down, and the least resistant are viruses with lipid envelopes. Okay, so it's just interesting to see the hierarchy of how antimicrobials affect those particular types of microorganisms. So we go with the type of microorganism. So if you think about factors affecting antimicrobial action, we've got the type of microorganisms, so prions are the hardest to get rid of. They can resist disinfection and autoclaving, which is not a good thing. But what you can do is, if you soaked that particular component or product in 1 normal sodium hydroxide for an hour, and then you autoclaved it for 30 minutes at 121 degrees C at 15 pounds pressure, then that will kill prions, or that should kill prions. Okay, so they are quite resistant. Then we go right down, and the last one, the least resistant are the viruses with the lipid envelope, because the lipid envelope itself you can target with antimicrobials. And as I said, you have to be aware of the starting population in a product or an environment. So if you have a very high number to start with, heat treatment is only going to decrease it a certain amount. But if you started with a lower concentration of or population, then the same heat treatment will knock out most of the population. So it just depends on what you start with. And we'll look at that here. So to describe that, you think you can describe it using microbial death rate. So if you look at a particular bacterial population and you subjected it to some type of antimicrobial action or mechanism, they usually die at a constant rate. Okay, so if you think about, so if we've got time on the x-axis, so we've got time in minutes, and just look at this, the pink line down in the graph, because this is the logarithmic decrease, because on the y-axis you've got logarithm of number of microbial survivors. Okay, so if you start with 10 to the 6 microorganisms, you heat treat it for a minute, you will decrease that population by 1 log. So you've got 90% of the population being decreased. Okay, you must remember this is a logarithmic scale. So 1 minute, you've killed 90% of the population. And then it takes quite a while to get rid of all of them. Okay, but you've knocked off a number of them, most of them, before, after that minute, with that minute. But if we start with a lower, so we've got the same, if we look at the graph on the right-hand side, we've got the same population, so 10 to the 6, on the same heat treatment, okay, but this is a high population load, a high microbial load. If we halve that population load, we gave it the same heat treatment, then you would get a decrease, so that 1 log decrease, and complete sterilisation by about 3 minutes rather than 6 minutes. Okay, so it affects the different, it depends on what the population load is to begin with. And also, if you're thinking about, so that's heat treatment, but if you think about chemical agents as well, if you have exposure to a particular chemical agent, if you just do 1 wipe, that might kill some of the, or might be a bactericidal effect on for some of the microbes there, but it might allow other ones to survive. So if you increase the contact time, then you will get an increased reduction of microbial load. And the same with heat. If you used a lower heat for a longer time period, that can actually help. That can be the same effect in decreasing the population. The state of the cells themselves, the microbial cells, if they are actively growing, so they're in that exponential growth phase, they are much more susceptible to antimicrobials. But if they've gone into the stationary and even into the decline or the death phase, and those bacteria have started to produce endospores, those endospores are very difficult to get rid of. So desiccation, etc., they can survive all of that. And then when they get into a nice environment, they can start to germinate and regrow. And also the environment. So some organic material can bind to the chemicals that you might use and stop them penetrating. So that's why I said you need a clean surface if you're going to disinfect a surface. So when you, if you're in hospital, particular instruments would be cleaned, physically cleaned first before they're sterilised, because, or disinfected, because you need to get rid of the excess organic material. Okay? So with the chemical methods, you need to think about the potency and the effectiveness. And to do that, you also have to consider the mechanisms of how they work. So if you think about a disinfectant, okay, so we're talking surfaces, are all disinfectants effective against gram-positives, or all disinfectants effective against gram-negatives? You have to work out how that disinfectant actually works, the mechanism of action, before you can say this particular surface is more likely to be contaminated with a gram-positive, therefore I will use this particular disinfectant. And you also have to think about the concentration of the disinfectant. So more is not necessarily better. Okay, so with some disinfectants, you need to have an aqueous phase. So you need some water in with it to be more effective. So if you think about alcohol, we used alcohol to, with our forceps to flame, to sterilise. Yes? But on the bench, when we were wiping down the bench, disinfecting the bench, what did we use? Did we use pure alcohol? No, it was 70%. So 70% alcohol is far more effective as a disinfectant than 100%, because you need that water in with it. And you also have to be aware that if an area is using the same type of disinfectant all the time, then you will get a build-up of resistance. Yeah? Because it's not always just killing everything. It could be killing some, but just decreasing the population of others. But the ones that are left will have a little bit more resistance. So you might be building up a population of resistance to a particular disinfectant. And that's why in various industries you have a pool of disinfectants that are used. So you cycle between them, so you don't get that build-up of resistance. The disinfectant has to be in contact with the microorganisms. And if you think about a porous surface, then the microbes might be able to get away, for want of a better term, from the disinfectant. The temperature also, if it's a low temperature, it might not be as effective as a higher temperature. So once again, you need to know what those disinfectants are like. You also have to think about the mixture of disinfectants. Okay? If you add two disinfectants together, is that going to be twice as good? Well, it may be, but they might actually negate the action of the other. So it's no, why even bother? They may also form together to form an explosive mix. So you have to be very careful about mixing disinfectants. And you have to be cognizant of how much time you're exposing the particular surface or microorganisms on that surface to the antimicrobial. So how do we tell if they work? So you've seen a lot of advertising probably, saying it's 99% effective against something. How is that worked out, or is it just advertising guff? So there's a number of different ways you can, and the disinfectant testing is quite rigorous, but there's a couple of different things you can use. So what's called the phenol coefficient. Phenol was, or carbolic acid, was discovered in 1867, and it was shown to be a very good disinfectant in those times. Okay, so now it was used as the gold standard. So we don't typically use a lot of phenols by itself these days, but we can use that to produce equivalent or ratios of the phenol, so produce the phenol coefficient. So typically that test is on Salmonella typhi and Staphylococcus aureus, and if the death rate is lower than 1, then it's less effective than phenol. If it's higher than 1, then it's more effective than phenol. Okay, so that is what the phenol coefficient actually means. You've got the use dilution test as well. Now this is quite interesting, and you can see in the image there's petri dishes with little cylinders, metal cylinders. Okay, so one of these tests is that the could be 10, 30, 60 little cylinders are filled with a particular concentration of microbe, okay, of a particular species. That is dried on that metal cylinder, okay. Then the disinfectant that is under test, you have different concentrations of that. You make different concentrations, and then you drop those little cylinders that have the dried bacteria on them, you drop them into this growth medium. The different concentrations of disinfectant. Yeah? Then after a period of time, it might be 2 minutes, 5 minutes, 10 minutes, you take that little metal cylinder out of the disinfectant and then put it into some growth medium to see if you can recover any cells from that cylinder. And you need, you'll look at the growth at the different concentrations to see how effective that particular disinfectant is. Everyone get that? It's a pretty neat way of doing it. And you can, bless you, you could also use viruses, spore-forming bacteria, etc., in those to coat those little metal cylinders as well, because some disinfectants say that they're effective against a particular type of organism, so maybe viruses or rhinovirus, and that way they can test for that. Another probably more direct way of doing this is of evaluating the effectiveness is the disc diffusion method. And so what you do here is you can see in these images, you've got little white discs on these plates, and then the growth around it, so that whiteness around it, is growth of a particular bacteria. So on the first one we've got Staph aureus, in the middle one E. coli, and then Pseudomonas aeruginosa. Okay, so they're grown as a lawn culture across or inoculated across the surface of an agar plate. Then you get particular discs that have been soaked in a particular or impregnated with a particular solution or antibiotic or something like that, and then you aseptically add that to the surface, and then you grow the plate. Okay, now with these substances that are impregnated of that little disc, they will start to diffuse out into the agar. And if they are bacteriostatic or bactericidal, then you'll have a clearing zone where the bacteria can't grow. So if you look at, we'll start with Staph aureus first, chlorine actually retards the growth quite well because you've got that zone of inhibition around it. E. coli, you've got a bit of a zone, but it's just something's happening there, but you still get a little bit of clearing, but not, if we look at the bottom side of it, not as much as you find with the Staph aureus. And then with the Pseudomonas aeruginosa, it's slightly effective, but there's not very much of a clearing zone. If we look at, say, a quat, which is a type of detergent, it's got quite a nice clearing zone there with Staph aureus, smaller clearing zone with the E. coli, and Pseudomonas just laughs at it. Okay, so it just grows basically over that disc. So this is a really good way of seeing very quickly how effective those particular antimicrobials would be. And you can, probably not on this one, but typically you would measure that zone of inhibition. So from the edge of the disc to the edge of the growth. So how do they work, these antimicrobials? They can change the membrane itself. So if you think about the membrane of a bacteria, it's actually there, a couple of different purposes. Inside the cell is it's got a relatively high osmotic pressure, okay, and compared to the outside environment. So if you have a cell membrane, then it's protecting it quite nicely. All good? Yes? But if you destroy that cell membrane in whatever way, you are allowing that breach, and so the osmotic pressures will start to equalize and you'll get destruction of the cell. And we'll see that a little bit later. And sometimes you can have some substances like some of the penicillins, they actually inhibit the production of the cell wall. So they can't produce the cell wall and therefore they can't survive. Some antimicrobials interfere with DNA and RNA synthesis. They might interfere with protein synthesis as well. So things like crystal violet will inhibit protein synthesis. Some of the antibiotics will interfere with the DNA and RNA synthesis. Okay? They don't have the genetic material, they can't replicate. Some will cause, with proteins, once again, you've got that lovely globular protein that you can see in A and B here. So the active protein, they call it. But if you destroy the protein, you destroy the bridges or some sort of stabilizing effect on that protein, they will unravel. Sometimes when they unravel, they will become inactive. Well, all the time they will become inactive. Sometimes though, they can regain their activity. But if you've got permanent denaturation, that won't happen. So they'll lose that activity. So if you think about a fried egg, okay? So if you look at an egg, a raw egg, that white part in a raw egg is quite runny. But when you apply heat, you will denature the proteins. You will make that protein inactive, and there's no way it can become reactivated. Okay, you've destroyed it completely. So that's permanent denaturation. So you think about the white of an egg. It's quite a nice one to look at. Then if you've got temporary denaturation, that means you've got the active protein being pulled apart basically to become inactive. But then if you take away heat for example, it has the ability to reform in that same formation. So it's only a temporary denaturation. So you can see with warmed milk, that's what happens. So there's a number of different ways that can occur. Then we've got soaps and detergents. Now, everyone remembers that a cell membrane is based on, what's the composition? It's a phospholipid bilayer, yes? The emphasis being on lipid. So if you have a detergent, then it's going to emulsify those lipids, yeah? And if you emulsify the lipids, what's going to happen to the cell wall? It's going to be destroyed. So if we think about who did their hands, swab their hands, yeah? Did much grow? I saw some fingernails where there was a lot of growth. So there was quite a bit of growth. So if you go to the bathroom and you wash your hand or you touch something and you want to get rid of what's on your hands, you go and use soap and water, okay? Soap is an alkali, so it can actually destroy microorganisms. Also, the physical rubbing of your hands together might actually dislodge some as well. And then you rinse them off with water, and probably you don't use the hand dryer. No, you don't. Did we see the hand dryer? Okay. Next time we do that, the collection of the air from a hand dryer, I'll put that image up on study desk for you, because there's a lot of things that grow after air from a hand dryer. Hand dryer. So, but you don't want to leave your hands wet. So what do? You can dry them. Okay? Why do you not leave them wet? Because microorganisms love to grow in a moist environment. Yes? And you can use other types of detergents. So other than for your hands, you've got cationic detergents for cleaning food utensils. So cationic just means that they've got hydrogen ions, and anionic for other cleaning things. So they're the negative ions. And yes, and these are the quats that I spoke of earlier. So these are cationic detergents, and they've got different structures. So they've got organic groups attached to their core, and they will change the plasma membrane so they start to leak. Okay, the bacteria start to leak the cytoplasm, and it will denature proteins as well in the cell, and that includes enzymes. So if you don't have enzymes working, then you can't replicate if you're a cell. It will be effective against gram-positive bacteria, fungi, amoeba, and enveloped viruses, but Pseudomonas, typically Pseudomonas aeruginosa, will grow in it. Pseudomonas aeruginosa can actually grow in 100% alcohol as well. So Pseudomonas is a pretty hardy beast. But these quats can be inactivated. So if you've got a lot of calcium deposits or magnesium deposits in that particular area, then the quat will be inactivated. We typically don't use a lot of them these days, but it's just so you're aware of what they are. Next group is acids and alkalis. We spoke about soap being an alkaline antimicrobial, but you can also have acids being antimicrobial. So if you think about lactic acid in bread, or benzoic acid in soft drinks, or sorbic acid in cheese, is that adding to the flavour? Well, possibly, but it's there for a much better reason. It's because if you've got an acidic environment, you're discouraging the growth of other fungi that might spoil that particular food or foodstuff. So how does it do that? It's actually really neat, because if you have acid in the environment, so you've got a lot of hydrogen ions, then the hydrogen ions will go into the cell, yes? And they will accumulate in the cytoplasm. And the cell is too busy pumping out those hydrogen ions to be able to grow. I think it's pretty neat. Okay. Heavy metals, and this is a lovely image here. So you've got a heavy metal and you've got a lawn culture, but you can see that clearing zone around it, around all of them. And here you don't have very much of a clearing zone. But why would you use heavy metals? So silver nitrate is used for gonococcal infections. So if someone is having a baby and they've infected with Neisseria gonorrhea, okay, that can be transferred to the newborn. Now, silver nitrate is typically put in their eyes because gonococcal infections can cause damage to the cornea and also will cause, if it's not treated, can cause total blindness. Okay, so some heavy metal compounds are quite good to use. Mercurochrome can be used on skin wounds, so it's an iodine, oh no, it's not, so skin wounds, but it can be toxic if ingested. Selenium sulfide, used for controlling dandruff. Background is it's effective against fungi and yeast, and therefore, and skin infections, and therefore you get it controlling dandruff. And copper sulfate is used to control algae. Now, have you heard of algal blooms, blue-green algae toxins? Yes? So if you have a large water reservoir, sometimes you might have algal blooms, and those algal blooms, the algae will produce toxins, and they can be neurotoxins or hepatotoxins, lots of different types of toxins. And you don't want that going into your water supply. Yeah? So there's a couple of things that you can do. You can spread the reservoir with copper sulfate to destroy that algal bloom, and obviously you don't know if you've got all the toxins, so then the water can be filtered through a charcoal filter to get rid of the toxins. Okay, so there's a number of different things that you can use. It's not one or the other. Sometimes it's a combination. Halogens can also be used, so you've probably heard of hypochlorous acid, and so the chlorine in swimming pools, yes? That's a great thing to use. So hypochlorous acid actually is used in swimming pools, in drinking water, etc. It kills bacteria and inactivates viruses, so that sounds like a really great thing, but it can be inactivated quite rapidly by organic matter, such as algae. Okay, so you have to think about the limitations. Iodine is used in surgeries, etc. It's a really good skin disinfectant, but it can be used in a tincture, which just means that it's mixed with alcohol, or as an iodophore. Okay, so it's combined with another organic molecule. Things like betadine destroys fungi, bacteria, amoeba and viruses. Excellent, except people can be allergic to iodine. So if they're allergic to shellfish, typically they will be allergic to iodine. Okay, so there's those concerns. With alcohols, we spoke about how they needed to be in an aqueous solution to exert their full effect. They can be used as a skin disinfectant, antiseptic, but it doesn't sterilize the skin because it evaporates so quickly. Okay, so that's why we rely a lot on our acid mantle, and we use PPE, etc. Phenols and phenolics, they will disrupt. So remember the carbolic acid that I spoke of, so the phenol coefficient. So as a disinfectant, phenol will disrupt the cell membrane and denature proteins, and with that inactivation, so they can also inactivate enzymes as well, which are proteins. Creosote, used to stop rotting timber, excellent, but it's a carcinogen and it's a highly irritant. Chlorhexidine is effective against a lot of microbes, so it can be used as a surgical scrub. So if you've been in a hospital, it's usually a kind of pinkish colour, chlorhexidine. Hexachlorophene was a great disinfectant, so Pfizer Hex. However, it's now been shown to cause neurological problems, i.e. you have quite a number of adverse effects on your brain. So it's not used now. Triclosan used to be in antibacterial soaps and hand creams, but because of its toxicity and its effect on the body, it's now banned. So while these are good, they have side effects that we may not know at the moment, but in the future we will know more about that. And that's why there's that constant testing. So number 7 is oxidizing agents. So you've probably all used hydrogen peroxide on a wound. Why? Why, you ask? Well, hydrogen peroxide, when you put it on a wound, it will actually break down and produce water and oxygen. And what can't survive in the presence of oxygen? Anaerobes. Yes, so any sort of wound that might be infected with, say, soil, you might use hydrogen peroxide because you're producing a lot of oxygen in that environment, so you won't get the production or the growth of anaerobes. Potassium permanganate, has anyone ever used this? It's like a purple solution that you can put on skin. It's great to disinfect instruments, possibly even skin, but if it's ingested, it's toxic. Okay, so you always have to think about how it's being used. And both of these, they oxidize molecules inside the cell. That's how they work. Number 8, I'm going to have to hurry up here. So alkylating agents, they denature your protein. Okay, so they also disrupt your DNA, and it may cause cancer. Some alkylating agents are actually used in treatment of cancer because of that effect on rapidly dividing cells. They insert into their DNA, so you get a dysfunctional DNA. Okay, so they can be used. They can cause cancer, but they may be used in treating cancer as well. Some of the aldehydes are effective because they can also kill off endospores. Okay, endospores are the ones that we really want to get rid of, and some of them can decrease toxins as well, which is what you would like. Now, I spoke about ethylene oxide before, and it's quite good because, oh God, it's got good penetrating power, but you can use it on substances that can't withstand heat treatment. Dyes can also be used to interfere with the DNA of the cell, and So some are used to clean wounds. Methylene blue can inhibit bacterial growth in culture. Crystal violet, as I said before, it's quite good because it can inhibit growth of gram-positives, and also it can be effective against some protozoa as well. And this is just a summary of the antimicrobials that just gives a good overview of what their uses are and the actions. So how do they actually work? And it's nice to know, with the acids, they obviously lower pH, but they denature proteins and they're used for food preservation. So if you see ascorbic acid in a particular thing, in a particular foodstuff, that's there to decrease the pH, okay, as a food preservative, even though it might say no preservatives added. So, everyone happy with that? So we've looked at why we need to control microorganisms, the growth of them. We've looked at some of the different types of methods to do that, and you understand that some microorganisms are more resistant to antimicrobials than others. So prions as opposed to gram-positive bacteria. Okay, so you need to understand that hierarchy of resistance, and each antimicrobial has its limitations. Okay, so it's really important to understand that, because even if you got something that was really filthy, lots of organic matter on it, and you dumped it in a bucket of disinfectant, it will disinfect the surfaces that are in contact with the disinfectant, but the surfaces underneath that organic material will not be disinfected. Okay? And some organic materials can actually combine with the disinfectant to negate its activity. So, practice exam questions. What do you think? Everyone's got an answer in their head? Will we check? Are you right? Okay. Well, we'll leave it there, and we'll come back with lecture 10.